All right. Hello, everyone. Welcome to this new edition of the Worldwide Neuro Forum. Today's guest is Debby Silver from Duke University. Welcome, Debby. The format is the same as usual. Little introduction. Debby will tell us about her work, and then we'll have some time for a discussion. So Debby did her PhD at Johns Hopkins University, finished in 2003. She was working on Drosophila, in particular on the role of JAK-STAT proteins in cellular migration. After that, she did her postdoc until 2010. At the National Human Genome Center Institute in Bethesda, where there she got interested in neural crest, but not so much, if I understood well, Debby, in the neural part of the neural crest, but some other kinds of derivatives, including melanocytes, and really started her mainstream interest in developmental neuroscience in mammals. From when she started her lab as an assistant professor in 2010 in Duke. So Debby's line of research is focused on the development of the neocortex, and in particular on two main axes of research. One that relates to the subcellular distribution of RNAs in different kinds of cells, and more specifically in progenitor cells. I imagine she'll be telling us a bit about that. And the second line of work relates to the regulation of cell cycle and the role of precise parameters of the cell cycles in setting cell fate in health and disease. So Debby, beyond her passion for neuroscience, actually was telling me a couple of minutes ago that she initially wanted to do marine biology, but she changed for neuroscience because marine biology was too imprecise. She had to go diving and look for shells on the seafloor, and each time she would go back down, the shells were not at the right place, and the net had moved. And she said, "You know what the hell? I'm gonna study the brain. It's gonna be more precise and simpler." So you'll tell us about whether you think that's actually the case at the end or during the talk. So Debby, it's great to have you here. The screen is yours. Looking forward to your talk. Are you talking?
Can you see it?
I can see it, yes.
Yeah. And can you hear me?
I can hear you. Yes, it's perfect. Yes, all is
okay. Good. Okay, great. All right. Well, thank you, Denny, for the kind invitation. This has been a really fun seminar series, and I think as we were just discussing, wonderful at this particularly crazy juncture in all of our lives for bringing all of us together. So I'm excited to have the opportunity today to tell you about some of the ongoing work in my lab. In my lab, we're interested in understanding how our brains are built. That is, how do we, actually, I'm going to turn on the pointer. All right, it's not giving me the option. That's okay. We're interested in understanding how our brains are built. How do our brains develop from this tiny structure at gestation week 3, for example, all the way to a complex.
So we are seeing your pointer, huh, Debby? We see it.
Okay. Okay, yeah. Great. Thank you. All the way to a complex, highly organized adult brain. And in particular, we're interested in development of the cerebral cortex, which of course is the structure of our brain that is critical for our higher cognition, analytical thought, etc. And it's also thought to be our newest structure to have arose during the course of evolution. And thus, by studying cortical development, we can hope to learn and gain new insights into aspects that make us uniquely human relative to our closest relative, the chimpanzee. So beyond getting a sense of development and evolution, by studying development, we can also learn important aspects about the etiology of diseases like autism, intellectual disability, or even like microcephaly, as illustrated by this MRI here. So how do we go about tackling these questions? Well, one of the main model systems that my lab uses is the mouse. As I'm sure you're aware, there are a number of differences between mice and humans. Our brains, of course, are gyrencephalic, whereas the mouse brain, as illustrated here, is mesencephalic. But one of the things that really struck me as a developmental neurobiologist is the amount of similarities. And you can see that over the course of development, the cortex is really expanding quite dramatically. And this is actually drawn to scale. And if we take a look at a similar stage to this E10.5, embryonic 10.5 day mouse embryo relative to a human fetus at this parallel stage, you can see there's a lot of parallel morphologies. So beyond this, of course, we know that there are a number of key cell types in common in the developing cortex, as well as key molecular pathways. And that's really one of the rationale for our work. So as Denny was just introducing, the lab has three main interests. The first is aimed, and these are not mutually exclusive, they're actually informing each other. But the first main interest is to understand mechanisms of cell fate and disease, to learn about both fundamental biology of development, but also the underlying etiologies of neurodevelopmental disease. We're also actively interested in understanding subcellular functions of radioglia, and in particular roles for subcellular localization and translation in this context. And then finally, we're quite interested in understanding genetic regulation of human cortical evolution. And today, due to time, I will be mainly focusing on these two aspects of the lab. But I hope, in coming years to be able to tell you more about our work on cortical evolution, as we have some really nice stuff brewing in the lab. So now let's take a step back and get on the same framework in terms of the cell types that we're going to be discussing today. So at the earliest stages of development, the cortex is composed of these yellow cells termed neuroepithelial cells, and these really serve the purpose of expanding the pool of progenitors in the developing brain. And over the subsequent days, these neuroepithelial cells are replaced by these green cells, the radioglial cells. Now, radioglial cells, of course, are essential for producing neurons and then eventually producing glia of our brain. And they produce the neurons, they can produce neurons directly, but the more common way in which radioglia will produce neurons is indirectly by making this population of transient amplifying cells termed intermediate progenitors. And of course, I'm simplifying all of this. If we were to move beyond a mouse, in a human, there is, in fact, many more basal progenitor cell types that are also critical for producing the brain, and I'll touch on those later. So over the course of development, these radioglia will generate neurons that are produced in an inside-out fashion, such that the neurons that are produced at the earliest stages of development, the deep layer neurons, are going to reside nearest to the ventricle, whereas the superficial neurons that are going to mainly extend connections to connect the hemispheres are located in the most pial regions of the cortex. And concomitant with excitatory neurogenesis, we also know that inhibitory neurons, which are derived from the ganglionic eminence, undergoing long migratory paths to reach the cortex, are then intervening and establishing connections that are critical for cortical circuitry. So I want to highlight a few aspects of cortical neurogenesis that will be pertinent for my talk, and also just really define the complexity and the dynamic nature of this organogenesis process. So the first aspect I want to highlight is that there is clearly dynamic extrinsic cues that both radioglia and neurons are exposed to over the course of development, and even within different spatial domains. So at the ventricle, of course, we have the CSF, and then at the pial surface or the basal surface, there's an overlying meninges. And then all of the cortex is, of course, being innervated by vasculature, as well as a number of transient as well as permanent migratory cells, like Cajal-Retius neurons, subplate neurons, and then, of course, the interneuron I mentioned earlier. And this is really nice work that has been illustrated by Alessandro Piani and others I've listed here. So beyond this dynamic extrinsic control of neurogenesis, there's also, we know, a great deal of signaling that's going on. And this is an area that I think is particularly exciting and one that we need to learn more about, which is the interplay between signals that are coming from the meninges, as well as neurons to shape and help define when radioglial divide and how they decide to make the right type of neuron. And then finally, another key aspect in terms of this dynamic control is the cell cycle. We know that over the course of development, these radioglia, their overall cell cycle becomes longer. And really pioneering work from L.J. Pilaz and Colette de Hayes' lab, as well as Calagaria and Huttner, has reflected critical roles for G1 phase and implicated S phase in duration in the potency of these radioglia to make neurons. So one of the key questions then is, how are these spatial and temporal events actually coordinated? And we know that there's a number of key signaling pathways, as well as transcriptional cascades, transcription factor cascades, that are absolutely critical for molecular regulation of cortical neurogenesis. But one of the things that struck me when I began my lab just under a year ago was the potential role for RNA regulation. That is, after the transcripts have been generated through transcription, what happens to them? And increasing genomic studies have revealed the importance of splicing and epitranscriptomic changes, RNA modifications, that are dynamically occurring in the cortex. And single-cell sequencing from many labs, including Denny Jabadan's lab, have revealed that progenitors themselves may actually be transcriptionally primed for different cell fates, that is, for the fate of a neuron or a progenitor. So this really argues that RNA binding proteins are uniquely suited to rapidly coordinate these gene expression and control how that transcriptome is actually made into protein in response to extrinsic cues. And this could occur, of course, at multiple layers: RNA localization, translation, splicing, and degradation. And I'll touch on a few of these today. Okay, so how might RNA regulation control development? How might these RNA binding proteins, shown here with these little cartoon blobs, bound to RNA, actually function? Well, we know one of the rationales for looking at these this class of proteins is that they're highly, highly expressed in the developing neocortex. There are actually hundreds that are strongly enriched in radioglial progenitors. Many of them in other systems have been linked to the cell cycle. Then what's really probably less well appreciated is that many RNA binding proteins act in autoregulatory loops where they can both influence each other as well as influence regulons of related RNAs to influence gene expression. So then our overarching hypothesis with all of this in mind is that by studying RNA binding proteins and more broadly RNA metabolism, it can inform unique aspects of cell fate specification and disease mechanisms. So today I'm going to build upon those ideas and tell you about two stories in which we've been studying this concept. So the first part of my talk, I'm going to tell you about mainly one RNA binding protein, and I'll tell you briefly about a second, that we've discovered have roles in cell fate and disease, and really that have helped us to better understand fundamental principles of cortical development. So beyond just learning about RNA metabolism, they've also given us new insight into how the cortex is built. And the vast majority of that work that I'm going to tell you about is published in recent publications from my lab. So second, I'm going to move on and tell you about our interest in looking at these very distal compartments of the radioglia, the basal end feet, and the basal process. And I'm going to tell you about subcellular functions that are happening there, in particular, roles for local gene expression in controlling cortical architecture. And the vast majority of that work is actually going to be unpublished work. Okay, so let's start with that first part of the talk. The complex that I want to tell you about today, and in particular the protein I want to tell you about, is a protein that's called Margot, highlighted here in yellow. And suffice to say, this is part of an RNA binding complex that is implicated in a number of aspects of RNA metabolism. But pertinent for this talk is the observation that mutations in a number of these components are clearly linked to neurodevelopmental pathologies like intellectual disability, microcephaly, as well as craniofacial disorders, which also co-present with neurological deficits. And over the past several years, my lab has generated mouse models in which we've manipulated levels of each of these components in the developing cortex. And as hopefully you can appreciate here, this results in a pretty significant microcephaly for haploinsufficiency for any of these results in a pretty significant microcephaly. And this is occurring via pretty comparable defects in neurogenesis. So I want to now focus in on this one mutant, Margot, and highlight for you what some of those neurogenesis defects look like, and then how we then took this further to better understand mechanisms. So the Margot phenotypes we discovered a number of years ago were really caused by defects in how in the right fraction, right types of cell fates that were produced. So we saw fewer progenitors, and here what I'm reflecting is fewer intermediate progenitors, as illustrated by TBR2 staining. We also saw concomitant with this more ectopic neurons that were made. Now, of course, when you have more neurons, you might think that the brain should in fact be larger. But what actually happens in this Margot mutant is that the vast majority of these ectopic neurons undergo cell death via apoptosis. And we were interested in digging into this further. And so again, a number of years ago, we discovered that when we manipulated levels of Margot, or actually in fact any of those other RNA binding components, it led to a really significant arrest of cells in mitosis. And this was true, as I'm reflecting here, in immortalized cells. Okay, so these defects of precocious neurogenesis and mitosis are quite common to a number of microcephaly mutants that have been shown throughout the literature. But one of the key gaps when we began the study was understanding how these mitosis defects may be actually integrated to ultimately influence these altered cell fates that we're seeing. And so LJP Laws, a former postdoc in the lab who actually now has his own group at Sanford Research Institute in South Dakota, wanted to tackle this question and asked the following question of whether Margot might control cell fate by regulating mitosis. And so for this study, he teamed up with another postdoc, former postdoc in the lab, John McMahon. And so here, what LJ decided to do was to use an H2B EGFP transgenic line to visualize mitosis in embryonic brain slices. Now, here you can see this is hours and minutes on the bottom, reflecting that the average mitosis of the control in vivo took place over about an hour at this particular age. Now, what he observed in the Margot mutants was a striking defect. That is, a large fraction of the cells had a significant delay in mitosis. And in fact, this particular example I'm showing here was delayed 2 to 3 times longer than it should be. But ultimately, over this 5-hour imaging period, it actually was able to undergo division and generate 2 new cells. So this really begged for us the question of what is the fate of these cells that were generated from this delay? And might this help to explain the altered cell fates that we had observed in vivo? So to tackle this question, John then developed an in vitro assay in which we could monitor cell fate by using either an antibody staining or using a transgenic mouse in which red fluorescence is indicative of newborn neurons. And he generated a number of movies like this. This happens to be an example of a neural progenitor that underwent division to produce 2 new neurons. And what he discovered was that at this particular stage in development, the control brains underwent primarily proliferative divisions at this stage. And in the mutant Margot brains, the cells that divided normally with normal mitosis duration also showed the same progenitor reflection of ratio of progenitor to neurogenic divisions. In contrast, hopefully what you can appreciate here is that the delayed Margot mutants showed a significant increase in neurogenic divisions. And this particular was biased towards asymmetric neurogenic divisions. So this is, of course, reflecting what we had seen in vivo, where more neurons were made. So likewise, also pursued live imaging to visualize the fates of these cells and whether they were viable or not. apoptosis was a phenotype that we had observed in vivo. And intriguingly, what he discovered was that under extremely long mitoses, the cells were preferentially producing cells that then went on to die. So they were non-viable divisions. So with this in mind, we wondered whether Margot might also control interneuron generation. So the studies I just showed you to date suggested that it was important for production of pyramidal neurons. But what about the cortical interneurons that are produced in these ganglionic eminence? And then really further, we wondered whether the similar mechanisms may be at play, and that perhaps Margot may control progenitor divisions by influencing mitosis. So this was work that was really pioneered by John. And what John observed was that in addition, so here I'm showing you an in situ expression pattern for Margot. You can see it's highly enriched in the radioglial progenitors, but it's also in the ganglionic eminence. So he devised a strategy where he could essentially fate map the cortical interneurons using NKX-CRE, which is a progenitor driver, and then using tomato to visualize the ultimate number of cortical interneurons present at the end of gestation. And for this work, he also teamed up with Charlie Sheehan, who really took a heavy hand in helping to complete this story. So together, what they were able to show was that the Margot heterozygotes led to a significant, about a 40% reduction in cortical interneurons. And you can see in the conditional knockouts, there is a virtual absence of cortical interneurons. Now, importantly, this was due to a defect in progenitors themselves. So when we do the same experiment using a DLX driver that drives expression or drives the knockout only in the migrating interneurons, we see no phenotype. So John then pursued this live imaging approach, now microdissecting ganglionic eminence and looking at those progenitors divisions. He saw that in a dosage-dependent fashion, mitosis duration was significantly increased. And I won't have time to show you this data, but it was published earlier this year. I want to summarize for you what the key findings were. The punchline is that the same mechanisms, at least some similar parallel mechanisms, seem to be at play. That is, in the conditional heterozygotes, the delayed progenitors have a preference towards producing interneurons at the expense of producing more progenitors. And then under the longer delay, we see a link with cell death. In the conditional knockout, there is a significant, and I should mention that this apoptosis is, we showed was p53 dependent. Now, in the conditional knockout, the progenitors that are delayed in mitosis, in fact, undergo what we referred to as mitotic catastrophe. So the majority of the cells, in fact, are unable to complete cell division and ultimately die. So in addition to reflecting a role for Margot in the subpallial structures of the brain, which we hadn't previously examined, this also highlights a role for cell cycle dynamics in cortical interneurogenesis. And in contrast to what we know about generation of pyramidal neurons, I think we know far less about how the cell cycle is integrated in with generation of cortical interneurons. So we think this will be interesting. So all of this then begs the question, is there a causal relationship between prolonged mitosis and altered cell fate? Everything I've shown you so far reflects a correlative relationship. And so to tackle this question, moving back to this initial study, LJ and John used two pharmacological inhibitors, nicotazole, which allows us to delay mitosis by depolymerizing microtubules, and STLC, which prevents the bipolar spindle assembly. It causes the spindle to transiently collapse. These are both reversible drugs, and we applied them to either embryonic brain slices ex vivo or to in vitro cells in culture. I can tell you that we saw no obvious defect in interphase, which reinforced our confidence in the use of these drugs. But what we did see was that at the longer mitoses, and these are excessively long mitoses compared to the control, which would be within this 60-hour interval, we saw that there were fewer proliferative divisions, more neurogenic divisions, and then, as you can see, a significant amount of apoptosis. So all of this then is paralleling what we had observed in the Margot mutant, in reinforcing then that there is a causal relationship between excessively long mitosis and altered cell fate. So Erin Mitchell-Dick, a graduate student who recently graduated from the lab, wanted to then ask, is this similar mechanisms at play with human neural progenitors? And so to tackle this, he used embryonic stem cells to differentiate into neural progenitors and then similarly exposed them to these drugs, was able to see that there was a longer mitosis. So to make a long story short, he saw similar patterns where there are more neurogenic divisions in the delayed STLC population, and in particular, more of these asymmetric neurogenic divisions and more apoptotic divisions. And so this is also published if you're interested in looking at it. So this altogether suggests that prolonged mitosis of progenitors is sufficient to directly alter what these progenitors ultimately become. We showed in our initial study that the prolonged mitosis can trigger an apoptotic fate in a p53-dependent fashion, but we were left with question marks about the mechanism by which this lengthened mitosis might induce a neurogenic fate. And in particular, we sought to understand and address this question of, does prolonged mitosis alter cell fate in vivo as opposed to ex vivo and in vitro? And what are the key mechanisms? So Erin decided to tackle this question by developing a new model in which he would use in utero injection to inject to STLC. And we coupled this with flash tag. This is a fluorescent tag that has been really pioneered in the cortical development field by Denny Jabadon's lab. And what it allows us to do is then to label the mitotically delayed progeny, mitotically delayed progenitors, and follow their fate over time. So we can then use this for all sorts of aspects to look at cell fate, but then also to sort out the cells. So Erin first established that this was working and showed that over a 3-hour interval after injection, he saw a pretty significant delay as quantified by phosphohistone 3. So about a 2 to 3 hour delay relative to normal. This was transient, which was also important for interpreting our studies. And then he asked, well, is this associated with increased apoptosis? And is this associated with altered cell fate? The punchline is yes. Again, I will refer you to his study that just came out last week. But essentially what we saw was that there is a significant amount of apoptosis in those flash tag progeny. There's also an increase in NeuroD2 positive cells and concomitant with that, a decrease in the radioglia progeny, the PAX6 positive cells. So we then wanted to take advantage of the system, having established the proof of principle, and ask whether if we were to isolate these delayed cells and isolate these brand newborn cells, can we learn more about what might be molecular pathways that are important for really driving and triggering cell fate changes in progeny? So here I'm showing you a graphic which represents a number of transcripts that we saw either significantly increased or decreased in these delayed progenitors. We did this both at 2 hours after injection and 8 hours after injection of the drug. So the first point that, of course, is always nice to see is in the particularly in the 8 hour or in this 9 hour time point, we saw known pathways that have been heavily linked to microcephaly mechanisms. And in particular, one of the most prominent pathway that was really altered was P53. But what this also does then is highlight new pathways and new transcripts that themselves actually may be regulated to functionally interrogate in order to better understand these the pathway from delay to cell fate changes. And this is something that we're actively interested in, and I hope the community finds it helpful as well. So altogether, what these studies have indicated is that prolonged mitosis may in fact be a mechanism to explain the etiology of at least some genetic and potentially viral causes of microcephaly. So we know that, of course, Zika virus infection in utero can also cause microcephaly. So in a microcephalic brain, we see, at least in the Margot mutant, and as well as in many others, that there is precocious production of neurons, apoptosis of some of these neurons, and together this is leading to a significantly smaller brain size and is associated with this delay in mitosis. Now, as I mentioned earlier, a number of microcephaly models published by a number of great groups have shown that that mitotic defects are evident in microcephaly models. So this concept itself is not new, but what was particularly new was that most of these studies have not used live imaging. And so the nature of how these mitotic defects actually might shape neuron production and neuronal survival was previously unappreciated. And there have been a couple of studies now that are reporting prolonged mitosis as a potential mechanism to explain etiology of microcephaly. And this paper from Arnold Kriegstein's lab actually used live imaging of organoids that showed that longer mitosis of outer radioglare, which are prominent in primates, in particular in humans, was associated with Miller-Dieker syndrome, a microcephaly-related disorder. So we're really excited about this as a potential mechanism, mainly to explain, and I want to emphasize, disease mechanisms. The role of mitosis duration per se in normal development is still something that I think needs to be explored. But we think that this may be a way that delayed cells respond, in that rather than making new progenitors, the default would be to take these stem cells out of the system, either via apoptosis or making a differentiated cell. So how might Margot integrate with other factors? Of course, you're probably wondering how Margot actually controls mitosis, and this is something we're very interested in. But more broadly, we wonder, do other RNA binding proteins show similar mechanisms? And really, given the vast number of RNA binding proteins that are highly enriched in the cortex, do other RNA binding proteins control cortical development by acting on progenitor cell cycle? And it turns out that when we carried out downstream sequencing analysis of Margot mutants, as well as the other components of this complex, we discovered a common dysregulation of all three of these RNA binding proteins in splicing of other RNA binding proteins. So basically, the punchline is, in a Margot haploinsufficient brain, you see mis-splicing of all the factors shown here with the string analysis. Now, some of these really caught our attention because we knew that they were highly expressed in the cortex, but there was one in particular that we got particularly interested in because of its disease relevance, and that is an RNA binding protein called DDX3X. This is a helicase and a canonical translational regulator. So Ashley Lennox, a previous graduate student in the lab, sought to really understand DDX3X mechanisms in the cortex. And this was, for this work she initiated, she worked later on with Mariah Hoy, who has continued to pursue these studies in the lab, postdoc in the lab. Okay, so why should you care about DDX3X? Well, it turns out that we think it's one of the most prominent genes that is linked to intellectual disability. Mutations in this gene are associated with 1 to 3% of intellectual disability cases. I want to emphasize this is an X gene, which is a whole nother layer of interesting aspect to it. These are de novo mutations for the most part. There are only just a handful of inherited mutations that have been found. So mutations in DDX3X are also linked to microcephaly. About 40% of kids with either nonsense or missense mutations exhibit microcephaly. And increasingly, we're seeing a clear link with autism. So we had a really productive and fun collaboration that we recently published with Elliot Scher's lab at UCSF, in which we expanded the number of mutations to 107 in DDX3X, and now he actually has additional mutations that he's discovered in patients with this with DDX3X syndrome. So this work was initially pioneered by Snyder's block, this 38 mutations, but then we recently published this vast number of mutations. And so our part in the study was really to get at the mechanism, and so we sort of serendipitously teamed up because we were working on these in parallel. And Ashley, together with Mariah, used CRISPR-based approaches and in utero approaches to transiently deplete DDX3X and ask about its role in progenitors, as well as to use the missense mutations, which are primarily found in the meat of the protein, the RNA binding domain, where translational regulation is really integral. She misexpressed these in cells looked at the phenotype there. And then finally, through some unpublished work, Mariah has been looking at conditional knockouts of this. So again, I don't have time to show you much of the data here, but I just want to show you, summarize how we think DDX is working based on these experiments. So the first is that DDX3X loss of function appears to result in an excess of progenitor number, particularly both radioglia and intermediate progenitors, at the expense of newborn neurons. And bringing this back to the same sort of concept of cell cycle regulation, Mariah has been using cumulative nucleotide labeling as well as live imaging, and her work is starting to really strongly the length of the cell cycle in these the etiology of these phenotypes. So stay tuned for that. But what she's more recently shown is that when she depletes DDX3X, specifically using a Cre driver in progenitors, not neurons, she finds that there is a massive microcephaly. So, and this really mimics what's seen in a number of the patients. So we're quite excited about these studies, which really argue that DDX3X dosage is absolutely critical for cortical development. So I want to now pause and give you an interim summary of what I've shown you so far. First, through the studies that we've pursued of Margot over the years, what we've been able to uncover is not only roles for this RNA binding protein as a master regulator of other factors and of cortical development, but really it's kind of taken us in our work in a new and surprising direction and revealed new roles for the length of mitosis in cell fate, particularly in the context of microcephaly. So where are we going next with this sort of work? Well, one of the key questions, of course, that we are interested in is how does this complex work together to control mitosis? And we can use point mutants that disrupt the components' ability to interact with each other to tackle this question of how RNA binding proteins influence mitosis and cell fate, and also using proteomics to better understand its protein partners in both progenitors and neurons. And that is work that is being pursued by a graduate student in the lab, Bianca Lupin. And then Fernando Alsina is together with some work from Bianca, has begun to discover some really exciting non-canonical roles for this complex in neurons. So stay tuned for that. So I mentioned that there's tons of RNA binding proteins expressed in the cortex. I showed you one example of how we were able to delve into this Margot network to learn more about one particular protein that we think is absolutely critical for cortical development. And we are now sort of like all in on the DDX3X analysis, and we want to better understand how DDX3X may be influencing translation in cortical development and disease. So Mariah is carrying out tour de force analyses of ribosome to better understand translational targets of DDX3X. Debby is a new graduate student in the lab. You know, this is, you can tell she's new because this is a post-shutdown image of her. And she's going to be really delving into how these missense mutations behave. We know from some work from Ashley that the missense mutations actually disrupt RNA granule integrity in the cell, and they lead to these ectopic aggregates in the cell. And so she's going to be looking at that further. Okay, so at this point, in the last part of my talk, I'm going to switch gears. And I'm going to now turn to the work that we've been doing over the past several years to better understand subcellular control of radioglia, and in particular, how RNA localization might be helping to shape function in these basal structures. So I'll remind you of the fact that the radioglia are highly polarized cells. They have an apical niche down here where the CSF is located in the ventricle, and then of course a basal niche where the meninges, the basal lamina, and then other key cell types are like interneurons and calretious neurons are located. So this process can be hundreds of micrometers long or even centimeters long in humans. And it's been, of course, shown to provide a scaffold for new neurons to migrate. And then it ends in these so-called end feet, which provide a barrier to prevent overmigration of neurons. When these end feet are not in physical contact with the basement membrane, neurons will actually migrate through the cortex. But interestingly, there have been a lot of ideas thrown out there by a number of groups, some of which I highlight here, that have suggested that these end feet may also serve as antennas to receive signals, some of which have been shown to be released from the meninges, like retinoic acid, or from upper layer neurons to influence neurogenesis. And this is something that's really, really an exciting possibility. And so this is work highlighted by a number of these groups that I've highlighted here. So radioglial morphology itself is also important, of course, for disease. So disruptions to proper morphology are linked to heterotopias, as well as lissencephaly, and that's highlighted in this review. So over the course of normal mouse development, the radioglia, I think one of the aspects that's probably not as well appreciated is that the basal structures become much more complex. You can see from an E14 embryo to an E17.5 embryo, the number of end feet that have formed it has increased, and then the number of processes has also increased. Now, what's really fascinating about this is that when Pashka Rakesh did similar analyses in the rhesus macaque, he noted similar extensive complex architecture in the primate, where you can see this is actually reflecting an end foot, and this is the basal process. So these really look very dissimilar to what we typically think of in a cartoon representation of a radioglia. So lots of protrusions that might be communicating with migrating neurons. And Eva Anton, as well as this beautiful movie that LJ made while he was in the lab, showed that these radioglia are highly, highly dynamic. They're not just simply static structures. So this is from an embryonic brain slice where you can see lots of protrusions being made. Okay, so lots going on in these structures. What do we know about them? Well, really the big question is, how might this morphology and how might this asymmetry of the radioglia ultimately influence cortical function? And it turns out we really have a dearth of an understanding of the molecular aspects that are at play in these basal structures. And so we really need a better understanding of the molecules and the events within these structures. So this was something that we were very interested in the lab, and we thought about this and kind of gained insights from thinking about the morphological similarities between a radioglia and just a simplified postmitotic neuron. And in particular, what I've reflected here with these little red dots is the striking role of RNA transport and RNA localization, both in has been implicated in both synaptic and axon outgrowth, wound healing, and other aspects. So given this similarity, it's perhaps not surprising that some RNAs have in fact been shown to localize to these basal end feet. Really older studies reflected that nestin mRNA, as well as an mRNA for acetoskeletal regulator ABA, are enriched in end feet. And I want to highlight a seminal study by Noriko Osumi's lab and carried out by Yuji Tsunakawa, who showed that cyclin D2, which is a canonical cell cycle regulator, RNA is highly enriched in these end feet. And through a series of elegant work in that study, they suggested that cyclin D2 may potentially be locally translated there in order to influence cell fate, where it could be asymmetrically inherited into the proliferative cell versus the neuron, which would be derived down at the cell body. Okay, so that was really our understanding several years ago of the list of RNAs that were localized there, but its role in radioglia was almost entirely unknown. And so we sought to test this hypothesis that RNA is actively translocated and locally translated in the radioglia. And this was work that was really pioneered by LJ and really heavily involved experiments by Ashley Lennox as well, both of whom I've introduced already. And together, along with other members of the lab, they were able to show that RNA is actively transported in the radioglia. And finally, once it reaches its destination in the basal end feet, it is competent to be locally translated. So we published this work in 2016. But one of the things that has dogged us, and so this is now 4 years ago, but for many years, is what is it actually doing up there? What is its function? So, so we got some clues from some really beautiful RNA immunoprecipitation studies that LJ had pursued, in which he had observed that the RNA binding protein FMRP was highly enriched in these radioglial end feet. So he used a GFP fusion protein with FMRP to pull out all of the RNAs that it was binding to in these end feeds. And what he discovered was that these were significantly enriched for certain classes of proteins, particularly cytoskeletal-related proteins and signaling molecules. Perhaps not surprisingly, given that this was using FMRP, we saw a large overlap with known FMRP targets as well as autism-associated transcripts. More recently, in order to really delve into the function of this, we also pursued a parallel approach to dissect out the proteome. And this is work that's actually unpublished and was pursued in a tour de force by Ashley when she was in the lab. So she took advantage of a biotinylation assay where you can introduce protein BRA, and in the presence of biotin, it's going to promiscuously label all of the proteins. So this has typically been used for fusion proteins where, let's say, for Margot, you want to figure out all its binding partners. Well, in this case, Ashley decided to do it with a cytoplasmic BRA and introduce it in by IUV, and then harvest out the biotinylated end feed and then pursue mass spec. So when she did this analysis, we saw a significant enrichment of, and you can use various thresholds, but essentially what we saw was a significant enrichment in particular of about 46 proteins with the classes shown here. So really mimicking what we had observed in the transcriptome and extending it further. So beyond the transcriptome, we also saw heavy constitution of ECM proteins as well and receptors. So this also what was nice about this was not only did we see significant enrichment of BRA itself in the mass spec, but we also saw enrichment of proteins whose RNA we had observed there previously. So RNA had come out of LJ's data set. And Brooke Darcy, a graduate student in the lab, is really interested in these and following up further, so stay tuned for that analysis. But really what I'm showing this to emphasize the idea that it led us to propose this idea and this hypothesis that local translation of signaling in cytoskeletal transcripts controls radioglial morphology, and at least that this is at least one potential function of local translation, and ultimately that this could impact how the neurons are organized in the brain. And so now in the last 10 minutes or so of my talk, I want to tell you about unpublished work. Hopefully it will be submitted onto bioarchives in the next week or so that LJ pursued while he was in my lab. So we wanted to test this candidate by, we wanted to test this local function by picking candidates that we thought would be appropriate for this. And we were, came about focusing on a Rho GTPase regulator, Rgap, called Rgap11A, which is required to dampen Rho activity. And we became particularly interested in Rgap11A through a particularly exciting collaboration with Frank Pelot's lab. And Frank's lab had actually, in parallel to us, been looking at RNA localization and had discovered that Rgap11A, I want to just pause here and say, for those of you who are familiar with Rgap11B, which is the human-specific form, this is the ancestral form. So Frank had shown that Rgap11A is enriched in end feed, and they had actually further deduced the localization element that was sufficient to drive its localization. So this was a perfect candidate for us to use to tackle function. Moreover, it's from work in C. elegans, Rgap11A is strongly implicated in cytoskeletal regulation. So disruption of Rgap11A would be predicted to lead to too much active Rho. So when we look at a mouse tissue section, and this is reflecting an in situ hybridization where purple signal reflects Rgap11A expression, we can see that it's localized at the PIA where the end feet are localized. And then Ikuo Suzuki and Pierre van der Hagen's lab actually did the same experiment in our collaboration in human fetal brains and showed that quite remarkably this localization is intact at the PIA as well. So arguing for us that this is not just some weird anomaly of the mouse, it's actually a conserved end foot localization. So when Yuji and Kamudi looked carefully at the RNA and the protein, and LJ has followed up on this as well, we discovered that the localization of both really coincided with these stages when the radioglia were becoming more complex. And I should point out, in work that was done both from in silico analysis as well as from Fernando Alsina in our lab, we found that its expression was specific to radioglia. And as I mentioned, Frank's group had identified the 5' UTR element of Rgap11A that's sufficient to drive its localization. I want to point out that this localization of the protein is evident both in the end feet, and you can see it actually making its way down the basal process. This localization into the basal process is entirely dependent as well on the 5' UTR. So we think that the protein may be actively diffusing back down from the end feet, actively or passively diffusing. Okay, so LJ pursued live. He used the MS2 system to indirectly label RNAs. This is from an embryonic brain slice. I'm showing you a chymograph over time, where hopefully you can appreciate there's RNAs that are moving up, and there's some RNAs that are moving back down to apical regions, and this is in the middle part of the radioglia. So this was exciting because it indicated to us that Rgap11A is actively transported. In fact, it's moving at speeds that are similar to what's happening in microtubule-based movements. So is the Rgap11A locally translated? So to tackle this, we actually were serendipitously, LJ discovered a serendipitous assay to peel off these end feet in which to peel off the meninges in which the end feet were retained and actually closed up. And this was something that we took advantage of for our transcriptomics and proteomics. And when he used this assay, you can see that the end feet are these intact green blobs, and then overlying this would be the basement membrane. So I won't show you the data due to time, but the punchline is yes. When he couples this assay, when he couples this assay with Dendra, we could detect that the RNA itself was locally translated in end feet. So the key of this, of course, is that it allows us to separate out diffusing protein from the cell body. So then I want to finish up by telling you what we think the function of localized Rgap11A is. So to tackle this, we used a method to sparsely label the radioglia using in utero electroporation, and then LJ carefully quantified the morphology in which the branch number, the more branches were reflected by more complexity. So a branch number of 4 would show 4 different branches versus a branch of 1. So depletion of Rgap11A led to two significant phenotypes that I want to point out. The first phenotype, and that's reflected in this tracing of actual cells, was that there was less complex branches in the end feet. But when we looked in the basal process itself, we also saw fewer branches. So there was less complexity in the intermediate zone region where the basal process of the basal process. So this is the knockdown. When he does an experiment blind, and I should mention this was all done blindly, adding back in the coding sequence, this doesn't rescue. However, when you add the coding sequence that contains that localization element, it is sufficient to completely rescue. Likewise, when we do a rescue experiment using a form of Rgap11A that is deficient for its Rho activity, we see no rescue. So I can honestly tell you we were so excited about this result because it was the first evidence for us that local translation matters and that it's particularly required for radioglial morphology, both in the end feet and along the basal process. So for those of you who don't really think about basal end feet the way I do and the way we do in the lab, you might be wondering why this might matter. Well, we carried out transmission EM and then were able to trace, and that LJ traced the individual end feet here in yellow. And this will reflect as this movie is proceeding, it's going to turn on its side, so you're going to see the peel surface, and these would be end feet here. But really one of the, forming the barrier as I introduced you to earlier, but really one of the striking things about this movie is that you can see that these end feet are not just in a vacuum. They're actually in very tight contact with other cortical cells, be it Cajal-Retius neurons, interneurons, etc. So we wondered then, might Rgap11A control neuronal migration? And Rgap11A expression is limited to progenitors, so we specifically wondered whether it would do so non-cell autonomously. So to tackle this question, LJ teamed up with Jing Liu, a postdoc in the lab. And so basically the question was, if we deplete Rgap11A specifically from radioglia using this one-day knockdown experiment similar to what I previously showed you, do we see altered distribution of inhibitory neurons? And do we see altered migration and distribution of excitatory neurons? So the first question, looking at inhibitory neurons, we noted no striking defect in the basal lamina, or to our best ability, we noted no significant reduction in calretin positive calretius neurons. However, there was a significant reduction in the cortical interneurons lining the marginal zone and lining the basement membrane. So we then decided to image the migrating neurons, and I'm almost done, just keeping a close eye on the time. And so to do this experiment, LJ used a dual electroporation parameter in which he labeled the neurons in green and then went back into the same brains, and one day later knocked down Rgap11A in the newborn, in the radioglia. And so in this way, we were able to disentangle cell fate from cell migration. So we're labeling neurons that were already born. And when he carried out live imaging experiments like this, what we discovered was that in the Rgap11A deficient radioglia, there is a clear defect in the ability of these new neurons to migrate along this radioglial scaffold. And that's summarized in this one graph, but we have other evidence of that as well. So finally, Jing sought to ask if this really matters for cortical lamination. And I'll remind you again that the basal process itself was less complex in the Rgap11A depleted radioglia. So to tackle this question, Jing decided to look at neurons that were produced prior to, their peak production was prior to when the radioglia were manipulated with the Rgap11A siRNA. I'm showing you ROR beta, but we've also looked at other laminar markers as well. So this is going to mark layer 4. And what we discovered was that at either P0 or even postnatal day 21, this transient knockdown of Rgap11A led to a significant non-cell autonomous disruption in the laminar organization of neurons. So altogether, this indicates that Rgap11A is controlling the organization of both excitatory and inhibitory neurons. And it brings me to the summary for this part of the talk, where I've shown you that RNA is competent to be actively transported in radioglia, where it can be locally translated. And we know that in the case of Rgap11A, it plays roles in local morphological architecture. And we believe that this then is having a non-cell autonomous impact on the ability of newborn neurons to organize properly in the cortex. Rgap11A, as I mentioned, is implicated in cytoskeletal regulation, so this does make sense. So altogether, this indicates that local translation in radioglia can impact organization of the brain. But the broader messages I want to come back to then are the following. The first is that what these studies have reflected is that these basal structures are not just passive, they're actually quite dynamic and highly regulated. In particular, are going to rely on local gene regulation to control their function. Second, I think this is going to help us to gain new insights that are relevant not only for cortical development of the mouse, but also for higher organisms. And in particular, these basal structures that I didn't really have time to get into today are really fundamental in a number of key progenitor populations that are most prominent in primates, like truncated radioglia, basal radioglia, and even PL astrocytes. And so whether these mechanisms are also at play in those cell types, I think is going to be really exciting to evaluate in the future. So where are we going next with this? So Brooke, who I mentioned earlier, is interested in understanding the transcriptome across development of these end feet. Lucas Serter, a new postdoc in the lab, is interested in understanding how translation is controlled. And Camila Musso is also investigating targets that we think are, another postdoc in the lab is interested in the targets that might be regulated and how signals might be regulating these pathways in end feet. And then finally, we're with Camila, we're also using human cortical spheroid models to better ask whether there are parallel mechanisms at play in human tissue. So to summarize, hopefully I've convinced you, if you weren't a believer before, that RNA regulation matters, and it can also highlight new mechanisms of cortical development, as I've described here today. So I pretty much thanked the people in the lab, as well as key alumni. I also want to make a shout out to Jing Liu, who is doing some of our work on cortical evolution. Wasn't able to talk to you about that today. And then I want to mention Carly Newman, who is sort of lab manager, mouse technician, who really helps keep the lab running. And then finally, the Rgap 11A story we could not have done without this really exciting and fun collaboration with Frank's lab, as well as members of his lab, as well as Pierre's lab. And then the DDX 3X story was through a collaboration with Elliot Scher that we're continuing to do today. And then the mitosis study was also done with Ted Salmon, who's a mitosis expert at UNC. So I will, I think I'm done. I will stop, and I'm happy to take questions.
All right, Debbie. Thank you very much.
So do I. You can stop my screening, I guess.
Thank you very much for this broad array of data and this nice integrative view of different aspects of cortical development. That was very interesting. Let me close this for you. Voila. Okay, that's good, I think.
Yep. We have parallel heads.
Yeah. All right. So there's, of course, a couple of questions. Let me open the window. So they're sorted by, this is too big, sorry. They're sorted by number of votes, meaning perceived relevance. So I'll go through them and we'll go through them together. So since cell cycle length increases during corticogenesis, would subtle changes in cell cycle length change the laminar identity of the progeny? For example, generate normally later or earlier born neurons?
Yeah, that's a great question. So with this in vivo paradigm that we recently developed, that was one of the goals, to evaluate whether we saw a shift in laminar fates by looking at later stages. So this is in the paper. Disappointingly, we didn't see anything. However, I think one of the ways that I'm thinking about this is that this was a really acute transient manipulation of mitosis, and it may be that if we found a genetic way to do this in a controlled fashion over, maybe 1 or 2 cell cycles, we might then be able to see a more potent phenotype. But I think it's a really interesting question.
Okay. For cell cycle manipulations to have an effect on fate, do they need to be in the M phase? What happens if you lengthen by the same amount of time, but in G1 or G2?
So that the G1 experiment has been done. Colette DeHay and Wieland Huttner's labs, LJ Pilas, who actually was in my lab, had done some of the G1 lengthening experiments. And actually, there have been additional follow-up studies on that as well. So if you overexpress cyclin D1 and lengthen G1, you will get more neurogenic fates. To my understanding, S phase has not been directly manipulated. I'm aware of studies that Wieland Huttner's lab has published in which he reports that longer S phase is also linked to a fate change of progenitors, but I don't think it's been manipulated directly as much.
I think there's a longer S phase in the ferret too, no?
Right. Yes. Thank you.
There's differences, specific differences.
Yeah.
Okay.
Exactly.
From Alessandra Pierani, great talk. Do you know if Cajal Retius cells regulate RNA or RNP localization at the end feet?
Yeah, thank you.
That's a biased question, Alessandra.
It is, but yeah, I mean, Alessandra and I have chatted about collaborating on this front for a while. We should talk again. We haven't yet tried to do experiments to manipulate Cajal Retius neuron number and ask whether we see shifts in RNAs going up there. and or translation. So one of the things that we're doing now is developing broader ways to monitor translation so that we're not reliant on reporters. And I think once we have those, we're going to be perfectly poised to do that. So I'm going to get back in touch with you.
Both Margot and DDX3X lead to microcephaly, but Margot loss of function have more neurons and less radial glia, while the DDX3X loss of function animals have the opposite, more radial glia and less neurons. Yep. Why?
So I think I didn't have time to show you the data, and actually some of this is forthcoming from Mariah's work. In the DDX3X, one of the other key aspects that's happening is apoptosis. So with our in utero paradigm using transient CRISPR knockdown at later stages, we didn't see much cell death. And of course there we are not monitoring microcephaly, but in the conditional knockout, we can see that there's a lot of apoptosis. So I would venture to say that there is going to be, we actually know that there's a similar shift in the knockout where there's more progenitors and fewer neurons, but this is also accompanied particularly in the knockout, conditional knockout, by a lot of apoptosis. So, I mean, the thing with microcephaly is it sounds very simple, but there's actually a lot of ways to get there. And of course, there's a lot of forms.
Okay. Does the effect of Margot also occur in other brain regions, and is it present during adult neurogenesis?
Yes, it's definitely in other brain regions. We shared these mice several years ago with a group who wanted to look in the cerebellum, and I think they found some interesting roles in cerebellar development. I mean, if you look in UrExpress or Allen Brain Atlas, you can see it's highly, it's a ubiquitous protein, but it's highly enriched in proliferative cells. And so then, not surprisingly, it is in the dentate gyrus as well. Yeah, that was something that was sort of on our list at some point to look at, and we never got back to.
Okay. Are there any changes in progenitor cell proliferation differentiation upon Rgap11A knockdown?
Right, so cell fate changes. Excellent question. We analyzed this after we did a 2-day cell fate analysis, and we didn't see any defect. So Rgap11A at early stages is much more highly expressed in the cell body. So there could be some roles for it in cell cycle and cell fate specification. However, in the paradigm that we used, which was a 1-day depletion, we saw no evidence of cell fate changes. So that was actually very important. So thank you for that question, because obviously you worry when you start looking at lamination that you're looking at cell fate. And we think, at least in this paradigm, we can exclude it.
Okay. Don't you think that the phenotype induced by the siRNA against Rgap11A might be due to global rather than local translation inhibition?
So it's certainly possible. We are electroporating it, but of course it might be knocked down in other cell types. I didn't show you the data, but when you look in when you look at human single cell sequencing data from Arnold Kriegstein's lab, as well as other single cell data from the mouse, and then qPCR that we've done, it is clearly only in progenitors. It's not in migratory neurons.
Well, I wonder if the question wasn't about global as in affecting the whole cell as opposed to the end feet or subcellular.
Oh, yes. Well, absolutely. So, okay. So certainly it could be affecting global phenotypes, and that's why the rescue experiments we did were really critical, because with the rescue experiments, we're able to ask about, okay, so if my hands are radioglia and this is the end feet, we're able to ask about how rescue of this protein composition up here impacts translation. Yeah. And so that the 5' UTR experiment we think addresses that.
Yes. Does longer mitosis impact the ability of a progenitor to stay well integrated in the epithelium and increase the chances of delamination, differentiation, apoptosis? Well, apoptosis we saw. What about delamination?
So Songhai Shi reported some defects in delamination that were linked to centriole loss that resulted in mitotic delay. So I think he had some nice data suggesting that correlating mitotic delay with delamination. In the Margot mutant, we've actually never published this. We do see more basally dividing cells. I can't say for sure whether that is due to a mitotic delay. I can tell you that we also saw, independent of the mitotic delay, so in a discrete cell population, we could see abnormal spindle orientation, and Fumio Matsuzaki's work has shown quite nicely that spindle orientation will redirect newborn cells to basal locations. So it may be the spindle orientation in that regard is driving that phenotype.
Okay.
And in fact, it becomes tricky for many of these mitosis defects because they may be affecting multiple aspects of mitosis, duration as well as spindle orientation, or even spindle size, which Colette DeHayes lab has also shown is important.
Okay. Very interesting work, Debbie, from Dana J. Huigol. What fraction of progenitors undergo normal versus delayed mitosis in the Margot mutant? Do you see normal divisions because of the penetrance/dosage differences of Margot mutation, or is there compensation by other factors with redundant functions?
Yeah, I love that question. So I think if I'm recalling correctly, it was a little less than half of the cells have a delay in mitosis. So that wasn't a haploinsufficient. And so why is that? Why could some cells be more prone to a delay? Or could it be that some progenitors have different levels of Margot? One of the things that we talked about doing and actually never went back to do, which this question makes me want to do, is single cell sequencing to get a better sense of how Margot levels are across different progenitors, because we don't know if that might be helping to explain it.
Okay.
Yep.
What happens to the N-feet and the localized Rnps during mitosis?
So I didn't talk about this, but during mitosis, there is the cytokinesis furrow forms in the cell body such that one cell will typically inherit the basal process. The cell that inherits the basal process and thus the basal end feed is more likely to become a progenitor. So Fumio Matsuzaki's work showed that, as well as some other nice studies. There is some interesting stuff, I think, going on during mitosis in terms of cytoplasmic transport in the radioglia, perhaps back down to a mitotically dividing cell. I mean, there's just a lot of dynamics going on in these structures that we don't know much about.
Mm-hmm. Maybe a final question, an intriguing one. Is end-foot structure uniform across brain areas? How about sulcus gyrus difference in gyrencephalic species?
Yeah, that's a great question. I don't know the answer. I think we'd have to carry out careful either EM studies and or immunofluorescence studies to tackle that. I think clearly there's radioglia all over the CNS, and actually there's a lot of really interesting studies that have reported parallel roles for morphology and influencing signaling and even synapse formation in radioglia outside of the cortex. So, but whether there's localization in for example, the retinal ganglion cells and other related cell types, I don't know, but I think it would be fun to look at.
Okay. Yeah. So Debbie, we've reached the end of the list and of the discussion. So thanks again for being here and sharing this with us. Thank you to all of you who are out there behind the screen. Next week it'll be on Thursday at 5. Our guest will be Sonia Garel, but the host will be Bassem Hassan. So I'll see you guys in 2 weeks, but I'll be following, of course, Sonia's talk behind the screen on the other side of the glass window. So have a good week until then, and take care. Take good care.
Thank you,
Bye-bye.
everybody.
Bye-bye.